It's a particular pleasure for me to have the opportunity to introduce our speaker today, my friend and colleague, Dr. Victor Karpiak. Because what you're going to see, I think people from SIM are used to seeing applications of genomics and multiple omics to cancer, to cardiovascular disease. And I think the point after Dr. Karpiak's presentation that will come through very clearly is that behavioral disorders and psychiatric disease, including addiction, are coming along very rapidly with the application of exactly the same techniques to those terrible diseases which we know in many ways are ravaging societies all over the world. We're delighted to have Dr. Karpiak here. His original training was first in the Ukraine, his native Ukraine, where he received an MD degree and then a PhD degree in psychiatry and addiction in Russia, before serving an internship at Maimonides in New York. And then we were fortunate enough to lure him to tropical southern Minnesota, where he did a residency in psychiatry, a fellowship in addiction, an addiction fellowship, and he is now the director of Mayo Clinic Addiction Services. I think you'll be fascinated to see what he and his colleague Joanna Bernacca, who's sitting right here, right behind him, have accomplished in terms of moving toward the application of the techniques which those of us in the Center for Individualized Medicine are applying very broadly, and in this case, applying it to alcohol use disorder, where he's going to, in the next few moments, describe the application of genomics and multiple omics to try and give us greater insight into both the understanding the pathophysiology and the treatment of these diseases. Victor, it's a pleasure to have you here.
Well, thank you very much for this kind introduction, Dr. Winchelbaum. As you said, I'm here to tell the esteemed audience today about our efforts to discover biomarkers for response to treatment of alcohol use disorders. My learning objectives are here. We'll start by defining at least we'll try to define the burden of alcohol use disorders compared to other neuropsychiatric conditions in the world and in the United States. Then we'll talk about the progress that we have made looking for biomarkers of treatment response to alcohol use disorders. And then we will discuss the relevance of those findings to the SIMS efforts to promote high-definition therapeutics and practice ownership in treatment of this group of patients. So let's start with the burden of alcohol use disorders in the world. As you can see here, alcohol use disorders are second only to major depressive disorder worldwide. There is a little bit of competition from the Alzheimer and other dementias in the developed countries. I'm not sure how long this competition will last, but remains to be seen. This is another interesting illustration compiled by my colleague, Dr. Nutt from the United Kingdom. So he compares in this graph the impact of alcohol use and alcohol misuse on individuals who are using and society compared to other addictive disorders. And as you can see, alcohol is much more significant problem. In the United States, this is SAMHSA, and as you can see here, it is about 14 million of Americans who are suffering from alcohol use related problems, including alcohol use disorders, and it's about twice the number of people in the United States who are suffering from other substance use disorders. To further put it in perspective, for you to see how it compares to what is nowadays the hot topic, which is problems related to use of opioids. As you can see here, in any type of comparison, whether you look at those who use or misuse alcohol or drugs in terms of substance use disorders, ER visits, or number of deaths related to those conditions, you see that alcohol use disorders have much more significant impact on life in the United States. If we look at the prevalence of alcohol use disorders and treatment utilization in the United States, this is a result of a very, very large survey which is conducted I believe it's every 7 to 8 years. So the last wave was completed several years ago, and it included several hundred thousands of responders. As you can see here, it's about 14% of individuals in the United States who meet criteria for alcohol use disorder every year. And if you look at lifetime diagnosis for alcohol use disorder, it is almost 30%. And of those, only approximately 20% are looking for treatment. And the perception is that this low level of utilization of treatment options is due to belief that treatment doesn't work. So clearly, we believe that evidence-based strategies are needed to improve treatment utilization. The question is, and throughout my study, whenever it will be an important question, I will have the picture of Uncle Sam because of the involvement of federal funding in my research efforts. So the important question here is that can we achieve this goal by developing efficient ways of matching potential responders with available treatment options, which is what we believe is the goal of personalized medicine. So what are the medications which are available so far? As you can see here, it's only 4 medications which are approved by FDA. And one of those is basically drinking deterrent. It's medication which scares people because they know that if they drink alcohol while taking antabuse, they will develop pretty bad toxic reaction. The actual medications directed at treatment of symptoms of alcohol use disorder, more specific craving, are only 3 medications, and 2 of those are based on the same substance, naltrexone. One is injectable, the other one is oral, and acamprosate is another medication. There are several other candidates, but neither of those reach the level when it can be approved by FDA for regular use. So what do we know about efficacy of acamprosate and naltrexone? It depends on the treatment goal. As you see here, number needed to treat to achieve prolonged sobriety with acamprosate is between 7 and 12, depending on different meta-analysis. While for naltrexone, if you're looking at the same goal of achieving and maintaining sobriety, the number needed to treat is 20. But if you look at other treatment goal, which is decreased alcohol consumption while still drinking, it is around 7. So let me ask the audience, how many of you think those numbers are, let's say, bad? How many of you think those are good numbers, promising numbers? So what I see is you really don't have any way to assess and answer this question. Well, let me give you a hint. So this is how number needed to treat for these medications compares to antidepressants, which are blockbuster medications. As you know, this is the most commonly prescribed type of medication in the United States. And the other blockbusters are, of course, aspirin and statins. So if you look what are the number needed to treat to achieve goals by using those medications, you will immediately see that the difference is not that significant. So how do we go about treatment? Well, this is a standard approach to treatment, which I'm sure those of you who are treating patients in other areas of medicine, you apply pretty much the same approach. You start by identifying that this is a patient who has particular type of disease, in this case, alcohol use disorder. Then you assess the predominant type of craving. As we discussed, there is different types of cravings which are being targeted by different medications. We'll talk about this more in the future. And then you discuss convenience, number of times medication needs to be taken, discuss side effects, then you end up discussing with the patient what is his or her selection, then you initiate treatments, schedule follow-up visits, address compliance, so pretty much what we do daily as physicians. However, we need to consider important challenges in this treatment selection and treatment results, and those are dependent on individual specific differences, including biological and psychological differences. There are also differences in terms of specific medications that we're using. They have different side effects, and they also are different in terms of their ability to achieve desirable effects. We also need to consider environment, especially if we're dealing with individuals who have behavioral problems or substance use. We need to consider their unique life circumstances. However, if we do have biomarkers which allow us to predict response, we at least can address the opportunity, and we can have an opportunity to personalize treatment selection based on matching between first and second aspect of treatment selection here. So with this in mind, S.C. Johnson, who was a long-term member of Mayo Clinic leadership, and in discussion with Dr. Mrazek, who was back then the chair of Department of Psychiatry, came up with this idea of setting up a goal of discovery of treatment response biomarkers, which will allow selection of efficient treatment options for each specific patient, which is the definition of precision medicine. And they came up with this definition in 2005. And they felt that the way forward is not to build another building, but rather to build a team of researchers which will be able to work and achieve this goal. So they thought that what they need is statistical geneticists, they need specialists in addiction psychiatry, psychiatry neuroimaging, and molecular pharmacology and experimental therapeutics, basic science. Mr. Johnson provided the initial funding, and it was followed by support from NIAAA in the form of P20 Center Development Grant, which was followed by the R21 grant a little later, and now we are supported by the combination of U01 and R1 grants, which I will describe in details how each of those efforts supported our findings and delivered the findings, actually. So the next question was, okay, we have a team, so now which medication we need to focus on? Or in other words, which probe do we need to use in order to understand its impact on treatment, and then try to understand what are the biomarkers and how we can understand more about treatment response. In discussion with Dr. T.K. Lee, who at the time, Dr. T.K. Lee is on the left upper corner here, who was a director of National Institute of Alcohol and Alcoholism at that time. He felt that although a lot of research was conducted around naltrexone, not much was done to understand the effects of acamprosate, which at the time was the market leader. It also known to support abstinence, as I mentioned earlier, which is the most common type of treatment goal that we hear about from our patients, and the most stable type of remission for alcohol use disorder. It is also known to suppress negative craving associated with negative emotions, the relief type of craving, which is again quite common in our patients. And as we already heard, It is known to help some patients, but not the others. So the clear question was, can we find biomarkers of response which were not known at the time? So we came up with 4 different projects as the different parts of our P20 application. As you can see, those are human and animal projects which are paired to each other. So we were looking for behavioral and functional markers of response to acamprosate in mice and in humans, projects 1 and 2. And 2 other projects were focused on search for imaging biomarkers, again, in mice and humans. And I had the privilege to lead the pharmacogenetic project, which was focused on search for variation in genes which are part of glycine and glutamate pathways, and check if those variations are actually associated with treatment response to acamprosate. We were able to recruit 443 subjects. Those who ended up in the study, we recruited more than that. We were focusing on treatment outcome defined as sobriety during first 3 months of treatment. And we were focusing on 36 candidate genes in those pathways. We also arranged with the colleagues in Germany who conducted another large study looking at efficacy of acamprosate, and they provided us with the data for 110 subjects for whom they have had both clinical outcomes and also genome-wide genotyping data. So we used this relatively small sample to replicate findings in our top 4 candidate SNPs. So this is the one of the important results of our study. Before we go forward to discuss clinical results and genetic findings, I want to show you that the levels of GGT, which is a common marker indicating sobriety in people who are drinking. So as you can see here on the left, before medications were started, those who ended up being abstainers on the left and those who ended up relapsing had similar levels of GGT in their blood. However, after treatment, you can see a clear difference between abstainers and those who relapsed. In abstainers, the level of GGT was much lower. This is a busy slide, but the reason I'm showing it to you is I want to emphasize the importance of looking at clinical characteristics of study sample and treatment outcomes before we do any genetic studies. As you can see, there are multiple characteristics which seem to be different between responders and responders. You can see there are numbers which seem to be below 0.005. However, as you can see that on the top, the strongest association was with the study site. And as soon as we adjusted for the study site, it was only 2 other variables which showed significant association. And those variables were baseline craving measured by pan-alcohol craving scale and the time between last drink and initiation of treatment with acamprosate. So we adjusted our data for those variables, and this is result of our genetic finding. This is a, I can say, mini Manhattan plot, which shows only results for SNPs that we tested in our study. And as you can see, 4 of those SNPs were much higher positioned than others, and at least 1, which is located in green to be gene, actually surpassed the level of statistical significance, and it survived even after stringent Bonferroni correction. We then tested the same result looking at slightly different phenotype, which is time until relapse, meaning how long people remained sober while in treatment. And again, you can see here that those with AA genotype, which is more common, had a longer period until relapse compared to those who had TT genotype, which is a variant genotype, and those with AT genotype were right in the middle. We then looked at results for association in the replication sample. Our SNP was not genotype, it was imputed in this sample, and as imputed SNPs are usually producing slightly weaker results, but as you can see here, it barely missed significance, but another SNP which is in strong linkage disequilibrium with this SNP was actually statistically significant. I also want to emphasize that, again, we were looking at some of the clinical covariates, and as you can see in this sample, the important covariants included intensity of depressive symptoms. And we'll get back to it a little bit later in my talk, why it is important. So we then looked at the potential importance of this particular variant, and as you can see here, it is part of the so-called E-box, or the sequence in DNA, which allows attachment of proteins which are responsible for transcription of a specific gene and a specific protein, I should say. While if A is replaced with T, which is a variant allele, this E-box is disrupted, which means that production of green 2B subunit of NMDA receptor will be lower, less efficient. Why does it matter? Well, as we know, the NMDA receptors which have B2B subunit in their structure are located outside of the synaptic cleft and are predominantly responsible for long-term depression, while the analogs NMDA receptors with predominantly 2A subunit are located inside synaptic cleft and mostly responsible for excitotoxicity and long-term potentiation. So the obvious idea here is that the change in balance of those two receptors in favor of 2A is usually expected to result in excessively increased glutamate neurotransmission related to this particular type of receptors. Dr. Fry conducted and completed his project where he was focusing on search for associations between glutamate levels in two brain areas. One is left dorsolateral prefrontal cortex, and the other one is mid-anterior cingulate cortex. And what he was able to show is that indeed there is a correlation between glutamate level and PAX score, and again, PAX is pan-alcohol craving scale, which is measuring intensity of craving. And that finding was in left dorsolateral prefrontal cortex, and in the second brain region, again, he showed that there is still a positive association between PAX scores and the level of glutamate, but even more interestingly, what he was able to show is that even before treatment, the level of glutamate at baseline is much higher in alcoholics compared to non-alcoholic controls. After treatment, this level goes down and pretty much reaches the level of non-alcoholic controls, while in other non-treated alcoholics, it continues to rise. Dr. Choi and Dr. Winchelbaum decided to look at correlation between treatment outcomes and glutamate levels in the plasma. And as you can see, they selected individuals from the same data set for whom we had plasma available. And what they found is, again, those with ability to respond to treatment, they are shown in red here. Before treatment was started at baseline, their glutamate level was much higher compared to non-responders, and it decreased after treatment with acamprosate for 3 months. And again, they did it in both discovery and replication samples. So what are the main findings of this stage of our research? We demonstrated that our efforts to identify genetic biomarker in this case green to be gene, as well as our efforts to measure glutamate levels in the brain and plasma, indeed showed statistically significant association with treatment outcomes, which means those associations can serve as justification for using of those findings as potential biomarkers, which is again consistent with the theoretical considerations which led us to choose those particular targets. However, as you can see in my next point, the effect size, the impact of each association on treatment outcome was rather small. For example, for the genetic variant, it was only 1.7%, which is clearly insufficient for practical use in clinical settings. So it became clear to us that if we want to achieve our goal of improving prediction of treatment outcome, we need to look at signature, which includes multiple biomarkers, and potential solution in terms of search for this type of signature was to look at genome-wide distribution of those biomarkers. And also we were hoping that use of the new techniques which allow to assess the combined impact of multiple genetic variants, in this case polygenic risk scores, may be helpful in this approach. We were lucky to have our ideas to be supported by NIAAA. We were funded to do a study which supposed to include data from our CETA cohort, as well as cohorts which were recruited into 2 other large studies focused on efficacy of acamprosate and naltrexone, Combine and Predict. However, I have to say that Combine and Predict were not designed to be a pharmacogenomic studies. So it was a secondary idea to include a collection of biological materials. And as a result, the quality of DNA wasn't as good, and we ended up with, as you can see, about 30% smaller, or 35-40% smaller sample than we expected. And as you can see, when you look at the line about outcomes, you can see that we are looking at different outcomes. So far, we completed analysis for relapse and time until relapse in those samples. And as you can see, there are some interesting findings here, but none of those is reaching genome-wide significant level, which is again likely due to rather small sample size. So the next question we ask is whether we can benefit from the use of more comprehensive assessment of those genome-wide data. In other words, are we able to find relationships between treatment outcomes and genetic liability, genetic ability to respond to treatment? And the way we approach this goal is we decided to train the method by using subset of our study data based on Predict and CETA samples, and then use these polygenic risk scores to see if it can predict outcome in the third sample, in this case, predict. And as you can see here, for us to develop this polygenic risk scores, we had to check different P levels. To make it a little more clear, what we are doing here is, depending on each cutoff point, we are including only SNPs above certain line. So 0.2 means all SNPs which are above this line. 0.1 is above this dashed line. And the most efficient ended up being the one with 0.05 cutoff. And as you can see here, indeed, it ended up showing that there is association between treatment outcome and this particular PRS score in the independent sample. The next question we asked if there is a way to assess treatment outcome based on genetic load for different conditions. What are those different conditions which, by the mean of genetic predisposition to those conditions, they may impact treatment outcome? And the obvious candidates were phenotypes related to alcohol use disorder. In this case, you can see alcohol use disorder itself. The next candidate would be alcohol consumption. Then we certainly were interested in the impact of comorbidities, including anxiety, major depressive disorder, PTSD. We also looked at insomnia and educational attainment. And what you can see on the right is there are quite interesting findings. We don't see much of association between treatment outcome and the actual genetic load for alcohol use disorder. At least it doesn't seem to be consistent between different samples. And there is a color coding for predict, combine, and CETA samples, and the black color indicates meta-analysis. There is also no associations with bipolar disorder, insomnia, or educational attainment genetic load. However, if you look at alcohol consumption, major depressive disorder, or anxiety disorder, you can see that each of those seem to be predictive of increased risk of relapse. Somewhat unexpected was the finding related to PTSD. As you can see here, in all three samples, we found that genetic load for this particular condition seems to be actually associated with resilience to relapse. We tried to recheck this association looking at slightly different treatment outcome, which is time until relapse. And in this analysis, it was only major depressive disorder which seemed to be predictive of relapse in all three samples. So what have we learned from our efforts to use genome-wide search for genetic markers associated with treatment response? Well, first of all, as I said, we have to accept that as of today, not enough studies were conducted which collected enough clinical and genetic materials in order to have proper power to assess the genetic load which may be predictive of treatment response. However, if we're using PRS or other more powerful techniques, we are able to see that there is a genetic signal. There is, in fact, evidence which indicates that genetic variation contributes to treatment outcome. The question is, again, how do we go beyond this modest effect size which we were able to achieve so far using just genetic type of biomarkers. Clearly, it becomes even more important for us to expand our search for other markers and look at comorbidities, look at relevant phenotypes within alcohol use disorder, and also look at so-called intermediate phenotypes like, for example, metabolomics or other omics, which are more proximal to genetic variation and in such a way may improve power for discovery of underlying biology. So we need to look at bigger picture. We need to understand why people do things. How many of you know this gentleman? Well, Dr. Winchellbaum remembers him. Well, this gentleman made his name by answering in a pretty famous way the question why he robs banks. This is a bank robber. He said, I rob banks because that's where the money is. Well, his next answer was actually more insightful. He said after the first answer, why does a smoker light a cigarette? And in fact, if you look at contemporary understanding of biology of addictive disorders, you can see that it is desire which drives binge intoxication, which then is followed by withdrawal and negative emotions, which again trigger desire to drink more and perpetuate the cycle. So what is craving? Craving is defined as a strong desire to use substance, in this case alcohol. It was reinstated in DSM-5 as a continuous measure which allows efficiently separate those who are dependent on alcohol from those who are independent within the continuum of alcohol users. And it can be measured in terms of magnitude, frequency, and it's also important to consider the contextual meaning of craving. As I said, it is important to understand whether people drink because they want to enhance their enjoyment of whatever they are enjoying, or they are drinking in order to suppress negative feelings related to, again, whether they feel bad because of physiological problems related to withdrawal, or because of whatever is happening in their lives, or because they have comorbid depression, anxiety, or other conditions. What do we know about relationships between craving, depression, and relapse? Well, as I said, craving is, especially negative craving, is known to be associated with desire to drink, to suppress negative feelings, including depression. Depression is also quite common in alcohol users, and especially in those who end up in treatment. It's up to 70% if you are looking at those who are treated in residential facilities like the ones that we have here at Mayo or at Hazelden, which is our collaborator. Now we're talking about it later. And it is known that depression, comorbid depression, is part of the reasons why people are not able to maintain sobriety long term. So again, Uncle Sam wants us to answer this important question: what are the mechanisms that are connecting depression, craving, and relapse in alcoholics? Actually, we did some research in this direction together with collaborators here at Mayo, Dr. Winham, who is on the left, and our collaborator from the University of Uppsala in Sweden, Dr. Bakalkin. We investigated relationships between craving, depressive symptoms, and history of depression. I'm emphasizing depressive symptoms and history of depression in individuals treated in our program. And we were focusing on candidate genes, prodynorphin and OPRK1, which are part of endogenous opioid system known to be associated with negative affect and dysphoria. So as you can see here, we indeed found that there is an association between variation in prodynorphin gene and alcohol dependence, as well as negative emotional states and time until relapse. But the interesting point that I want to emphasize here, and it is color-coded, as you can see, each of those associations is driven by either male or female parts of our study sample. More specifically, alcohol association was associated in males. Prodynorphin variation was associated with alcohol dependence in males, but not in females, while in females, variation in this gene was associated with shorter time until relapse and tendency to drink to suppress negative emotions. We then decided to replicate this finding, and we did it in collaboration with Dr. Ulrich Preuss, who at the time was working in University of Munich. And he had much smaller samples, so not all results reached stringent level of statistical significance, but you see the same type of pattern. Again, alcohol dependence was strongly associated with prodynorphin variation in males. Emotional discomfort as a reason for drinking was associated with prodynorphin variation in females, while longest period of sobriety was again associated with variation in males. So what have we learned from those candidate gene studies in both CETA and prodynorphin variation line of studies? Again, we got a proof of concept that genetic variability is associated with negative craving, treatment outcomes, and depressed mood in alcoholics. And that's similar to other disease, complex disease phenotypes. It is likely that multiple genetic variants are contributing variables, effect sizes to this genetic load. So again, it emphasizes the importance of us using tools which allow to assess this combined genetic load, which is similar to a polygenic risk score. So again, the important question here is how exactly genetic load, clinical variables, and demographics, in this case, sex, impact phenotypes related to alcohol use disorder. So in the next study, we again went back to our CETA sample and we asked the next question: is there a difference between the impact of emotional states, like for instance, tendency to drink during negative emotional states, versus diagnosis of major depressive disorder on alcohol use? And before we go into results, I want to show you again the difference between males and females. As you can see here is something that you pretty much know very well, right? Males tend to drink more, but females tend to have more comorbid mood problems if we're talking about alcoholic females. And also, as you can see here, is that females more commonly report tendency to drink in negative emotional states. So, however, what we discovered in terms of the impact of lifetime versus current diagnosis of depression, those seem to again impact alcohol consumption in males and females in a different way. In females, again, it's color-coded, and I don't need to explain you the choice of colors, right? Those of you who have children will be able to follow my line of thinking. So in females, you can see that it is current major depressive disorder which is associated with increased average number of drinks per drinking day and the maximum number of drinks per drinking day. However, in males, the effect is different. And it is different not only in terms of direction, but also in terms of the impact on other drinking characteristics. They seem to drink less often, and they seem to have less of heavy drinking days. This is another way to show this association. Again, if you look at the top graph, you see that males without MDD history have this clear association. The worse their mood, the more they drink, right? On the other hand, males who have a history of major depressive disorder, which is not present at the time of our working with them, they don't have this association. It disappears. And this is not the case in females. In females, they drink more when they are in bad mood, and this is regardless of whether they have or don't have history of depression. This is to emphasize the next point. The impact of male pattern of drinking is not related to the amount. It is related to the number of drinking days, which means there is likely something which is triggering their desire to drink, and they seem to be less sensitive to this trigger being negative emotions if they have history of major depressive disorder. So what have we learned from those studies? First of all, I think very important finding is that state-dependent factors and trait-dependent characteristics impact alcohol consumption in a different way in alcoholic men and women. Emotional states tend to increase per day consumption in both males and females, while lifetime history of depression, which is a trait marker, seems to decrease the number of drinking days in men. So the important question is, can genetic variability explain those differences? So again, we went back to our sample and our data, and what we found here is that, yes, genetic predisposition to major depressive disorder in men is associated with lower total number of drinks, while in females, the effect seems to be opposite, although it is not reaching statistically significant level because we have much smaller number of female in our sample, but again, effect is in terms of increased consumption. What is shown here on the right is the results of interaction test, and it allows us to see this difference even more drastically. As you can see on the right is where most female drinking consumption measures are, which means presence of genetic load for major depressive disorder is increasing the alcohol consumption, while in males, which is on the left, presence of genetic load for major depressive disorder is decreasing measures of alcohol consumption. Now, what was the most interesting and most unexpected is the last point. When we adjusted those findings for a clinical diagnosis of major depressive disorder, the findings didn't change, which means that effects of clinical diagnosis and genetic load, although they go in the same direction, they seem to be independent. And this is a summary of what we have achieved so far in our CETA research. We have shown that biomarkers associated with alcohol use disorder and its treatment can be identified. So we achieve our goal of proof of principle. We identified limitations of previous studies, which are mostly related to small sample size of those studies and lack of proper collection of biological samples. And we have demonstrated importance of using advanced techniques which are able to identify polygenic risk scores and accounting for clinical and demographic characteristics as important factors. So where are we going with this and how are we planning to get there? Our goals are listed here. Goal number 1 is discovery of high-resolution signatures reflecting effect of existing medications on disease-specific phenotypes. Goal number 2 is discovery of multi-omic signature reflecting patient-specific disease phenotypes. And the goal number 3 is development of efficient algorithms for matching between 1 and 2. Goal number 4 is discovery of novel treatments based on those multi-omic signatures, and goal number 5 is implementing those into clinical practice. This is our roadmap. So in order to achieve aim number 1, we plan to collect biological samples allowing multi-omic work. Goal number 2, to conduct these studies of high-definition phenotyping in subjects who are treated in real-life circumstances. Then we plan to use artificial intelligence to deal with the huge data sets that we expect to be needed to deal with, and then use patient-specific cell lines and organoids to uncover biology and hopefully restore function to the normal level, and then assess the impact of those novel treatments in real life. And I'm happy to report to you that future is pretty much here. In fact, we received support from NIAAA to conduct 2 related projects, one looking at pharmacogenomics and the second one looking at pharmacometabolomics of acamprosate treatment response. As you can see here, those are really parallel projects. We'll start by identifying genomic and metabolomic markers associated with treatment responses in already collected data set. Then we will apply the same approach to identify biomarkers associated with treatment response in new data set of patients who will be randomized to treatment with acamprosate and placebo, which will allow us to see effects which are medication specific and medication independent, but still associated with treatment outcome. We will then apply collected data to focus on functional meaning of those findings, including use of iPS cells, which are specific and collected from our study participants. And this is our research team and collaborators from Studies Predict, Combine, NIAAA, and Hazelden Betty Ford Foundation. And thank you very much for your attention.
Thank you for participating. Please click the button below to complete the evaluation and obtain credit.